So welcome everybody. Thank you for joining this first episode of the webinar series of Ecogen. We are just gonna welcome you and explain the house rules. After a few years of online meetings, I think we already are familiar with the typical house rules. We can have the next slide. Just as a reminder, please, if you're not speaking and if you don't have your hand raised, mute your microphone. If you have any question or you want to interact, please use the feature of Zoom to raise your hand. And as you already heard, we are recording this session and it will be available online. So please, if you don't want your image to be used and to be shared online, deactivate your cameras. Today we have Ana Granados chairing this session. Ana is the director of EFAB and the Secretary General Fabret TP. So what EFAB is doing will be explained in more detail later on, but ideally...
Mas quanto é que precisa para eu perceber?
But ideally, FAB is representing the breeding companies and breeding organizations in the policy environment in Brussels. And we are really glad to have Anna introducing and explaining what is the main aim of the Ecogen cluster. So I will leave the floor to you, Anna.
Yeah, thanks, Remark. Good morning, everyone. So as Remark said, we are here today to talk about 3 European projects, which are HoloRuminant, Rumigen, Geronimo, 3D omics, and livestock. They have made a joint initiative of collaboration and communication of what they are doing. And I'm very happy to host this session today. So the introduction will be made by me. As Remark said, we are an organization working mainly in Brussels at the European level. We have 2 organizations mainly, and one representing the private sector, and the other one is a research and innovation technology platform. So it's something that is recognized by the European Commission, in which we put together research from the private and the public sector. And this is why we are involved in so many European projects or interreg initiatives that you can see the logos here. And 3 of them are part of this Ecogen cluster. As the Remark said, our work is to make lobby at the end of the day. So we promote animal breeding and genetics in Brussels, but also research and innovation. So that's why we want to put today both this together. They are sister organizations. So whatever it is for priorities in research and innovation, or to facilitate the work of private sector in the field, this is conversations we are having with the European Commission and the Parliament. The next slide is just about the logos of the organizations we are representing, so I will not spend time on this one. And I will go to the next, and why these projects we are going to talk about today and the collaboration between them is important also for our work. As you know, animal breeding is, there is an animation that should not work. Okay, here it is. It's a combination of many different characteristics or many different traits. This way of performing breeding programs and putting all these things together to choose the next generation of animals has changed a lot in the last years. This is the way we explain to policymakers in Brussels. So in the 70s, the 80s, because we have to choose a period, it was in a way that the focus was on production traits. And since the introduction of genomics and further knowledge in genetics and many other things, of course, we have moved to a more balanced approach with many different traits that are what we call the 6 pillars of our code of good practices, what is called FR. So So we need more research to improve this balanced approach and to introduce further traits in breeding programs. And this is why we are here today. So these 6 pillars, of course, are completely different depending on the regions, of the production systems, of the farmer choices, of the availability of resources, feed, but not only, other ones too, and also the social, environmental, and economic situations of farmers. Code FAVAR tried to respond to this. Code FAVAR is this code of good practice with the 6 pillars I mentioned. This is not something we ask our members to do. It's the way our members are already implementing in the field these 6 pillars. So again, we need further research and innovation. And yes, sorry, I don't know why these animations are not going in the right way. And animal breeding tries to address the 3 pillars of sustainability and improve animal health and welfare. So here are some examples on how animal breeding can tackle some of the challenge of the terrestrial livestock sector. In this case, because we also represent aquaculture. And this is just the current situation. So again, Ecogen can respond to improve the balanced breeding approach and adding additional breeding objectives and traits to the current breeding programs. So here is Ecogen. Again, this is not a project, it's a group, it's a joint initiative of 5 different projects. You are going to hear from all the coordinators, and the key focus areas are on how to develop sustainable assessment of the livestock sector, how we develop an assessment of the sustainability of the livestock sector and make more sustainable practice. There is a lot of animal genetic research. That's why we are here. There is not only, of course, but there is a lot of animal genetics. And the purpose is to utilize advanced technologies, investigate microbiomes that will be very well explained by the coordinators, and to take the advantage of having a holistic approach altogether. So I'm going to give the floor to the speakers very soon, that they could give more examples and explain how they tackle all these issues. Different, but all together. The 5 projects are European funded projects under Horizon 2020 or Horizon Europe. So we have more than 100 partners and more than 50 countries, including some third countries. Of course, some of the countries are represented or working on more than one of the projects of today. And here are your speakers of today. Diego Morgavi, explaining HoloRuminant. David Yáñez, explaining livestock. Frédérique Pitel from INRAE about Geronimo, what is focusing on monogastric only. Rumigen will be represented by Pailhoux, his coordinator, and Antton Alberdi will be explaining 3 omics. So without not spending more time and aiming to listen to the scientists, I will give the floor to Diego, because I forgot to say that I've stopped sharing, that this joint initiative is led by HoloRuminant. So this is why he has first the floor. And Diego, can you share your screen, please? Okay, super. The floor is yours.
Oh,
yes.
Okay.
Thank you very much.
Thank you, Anna, for the introduction. I'm Diego Morgavi from INRAE in France, located in Clermont-Ferrand. Within the whole 5 projects, the HoloRuminant is on the same call as 3D omics. HoloRuminant is on the ruminant side, and 3D omics is more on monogastrics. So the objective of this project is to understand microbiomes of the ruminant holobiont, so the consideration of the microbiomes and the host animal. And the goal The goal of the project is to elucidate the role of microbiomes and how they interact with the host animal. And we are looking on this interaction between the microbiomes and the host animal from birth, when the animals are born, and throughout fundamental life events like could be weaning, transition, up to adulthood. And we want to characterize how these microbiomes establish, how they change over time. We are determined how they connect between them. So, of course, in ruminant microbiology or microbiomes, the gut microbiome and the rumen is very important, but we are also looking at other sites in the gastrointestinal tract and also in the upper respiratory tract and the relations that can be with other microbiomes like the milk microbiome. We want to know whether these microbiomes are heritable, whether they can be transferred from the mother to the offsprings, and of course, how they influence the host in several ways. In what we call key performance indices is the production, but it's also in other indices that could be health, can be welfare, among others. So the aims of the project is to understand the role of microorganisms in livestock production and how they influence the animal health, productivity, and the overall sustainability of the production system. And the result of the project, we hope that they will be used to develop diagnostics, management, and nutritional strategies to improve the environmental efficiency of the production. So in the project, we have 25 partners represented in 17 countries. Of course, it's an European project, so most of the partners are from Europe, but also we are lucky that we can have other partners from other parts of the world, so we can relate our results and compare our results with these partners. The research is organized in different activities. So you can see the 4 main points in activities, in research activities, but also we are looking into the perception of consumers vis-a-vis of not our research. Also the knowledge of consumers on microbiomes and the knowledge of farmers on that, and whether there are what we are going to propose in the future are acceptable both to consumers and farmers, and also it's important to evaluate the possible adoption of the technologies that we are going to develop. It's also, I want to emphasize that this is a project which has a TRL of 2 to 4. So for those which are not familiar with that, it's more on the basic side. We are trying to understand and develop technologies, and our results will be used later on to the application at the field level. So for those 4 main activities, we have one activity that is consolidation, of existing knowledge. We are not producing knowledge that is already there, but we are gathering that and consolidating that knowledge. in both in the meta-taxonomic studies and also all the information that is on genomes of ruminant-associated microbes that are either cultured or uncultured. At the same time, we are producing new data, and that new data is going to be linked to be used to link these older meta-taxonomic studies and the studies that are still developing in the meta-taxonomics area, and so we can relate that to function. Another activity that we have is we want to understand the causes, the effects, and the stability of the microbial communities when they established in different body parts, in both in cattle and sheep. And for that, we are doing a large longitudinal study in different places. We have 8 sites all over Europe in 7 countries, so we can compare also that the effect of the acquisition of the microbiome in different situations. And also we have more focused studies to establish the link between those host-associated microbiomes and phenotypes in health and disease, and what are their effects on welfare, how they affect production. So all that leading to improve sustainability of farm ruminant. So the information that is going to be produced in these 2 activities is going to fit the consolidation one that I talked to you before, and is also fitting on another activity that is the integration of these microbiomes, that using not just genomic information, but also a multi-omic layer of information. We are using transcriptome and proteome, as well as the genomic information of the host. So that will be used, that integration, to better understand what is happening, and not just for the objective of this program, but can be used for other research objectives. So the nucleus of the project, it can be summarized in this figure, where we have existing and new information that is consolidated. We are developing tools also of integrating this information, and at different life stages of the animals, from birth up to production. And we have, like, as I said before, we are going to start to study whether there is any link between multi-site microbiomes. There's still an open question whether the rumen has a link with like the upper respiratory tract microbiome, whether there's a link between the milk microbiome and mastitis and the rumen, and so on. So we are going to have better answers on that. We are going to study these microbiomes within one site, like the rumen, but also how they interact with other sites. And also we are looking, at least for the rumen, not just on the bacterial part of the microbiome, but also we are looking into how is the interaction with the archaea, which is so important in the case of methane production, and also anaerobic fungi and protozoa, which are often overlooked in the studies. We are doing that using a holo-omics approach, as I said, multi-layers of omics technologies, and also combining the microbiome with the host genome. We are going to get better information on how this microbiome is transferred from mother to offspring, if they persist, how they persist, how stable they are. And we are doing that in challenging life periods. So we are looking a lot on the focused studies on the effect of the mothers, the effect of weaning, the effect of a feed transition, for instance. And within that context, how they affect the weaning, the effect of the mother, the health and welfare of calves and lambs, how they affect the diarrhea or respiratory diseases, for instance. And we are doing that within a diversity of production systems. So we are hoping that our results can be used in different contexts. As I showed you before, there are several sites, and the studies are done in several European countries under different conditions. And last but not least, we are also looking into the environmental efficiency in methane production, for instance. So all that leads to what I call improving the environmental efficiency of animal production. And that is where the ecogene idea of having a cluster of projects on the same idea comes together. Just to finish, a few questions that are for our fellow projects and also open to the audience that working in research. Could the ruminant microbiome be linked to some of the phenotypes or genotypes that you are observing? Because we are developing also tools that are going to be open access, and the tools and all the data set that we are producing can be used, I think, and I hope, in other studies. It's a question mark, but it's more an affirmation, I think. I think also they can be used to, if you are a private partner and you want to develop a product, can be used for that, and can be used for policymakers or people that give advice to farmers as recommendations. And with that, I will finish. Thank you.
Yeah, thank you very much, Diego. So more fundamental research project. So on this cluster of 5 projects, we have different levels of research, and HoloRuminant is a good example of a more basic research, and other ones will be more not more focused, but they will have a focus more on implementation in the field. We can give the floor now to Eric Pailhoux from Rumigen. So as the name makes understand, there is another project focused on ruminants. And we can see your screen, Eric, so you have the floor for the next 12 minutes. Thank you.
So, good morning to all, and it's a pleasure to present you the Rumigen project. So Rumigen stands for Toward Improvement of Ruminant Breeding through Genomic and Epigenomic Approaches. So next slide, please. So the main aim of Rumigen is to improve the selection scheme for the dairy breed, for mainly the dairy breed of cattle, and for breed, of course, that are managed by genomic selection, because we want to improve the selection scheme. So our main objective is to improve quantitative genetic tools in different bovine breed, as I said, and we are mainly interested by Holstein's breed, but not also. And so we want to add through the addition of new traits of epigenetic data and potential use of new breeding technique. And in BT, in this case, stands for mainly for genome editing. And so the new traits that are under investigation in Women are genetic diversity and also adaptation to new environment and mainly to climate change and to try to improve efficiency, sustainability, and resilience. And also in this Women project, we have an important level on social science in order to try to evaluate the societal acceptability of breeding strategy in general, mainly also on NBT and genome editing, but on breeding strategy in general. And our aim is to try to co-construct new breeding scheme with the society. So next slide, please. So very briefly, this project has been obtained in the call in 2020. The call was named Genome and Epigenome Enabled Breeding in Terrestrial Livestock. And in this call, there have been two projects that have been financed, supported the Ruminant project on ruminant and also Geronimo on monogastric that we will hear just after Rumigen, I think. So it encompassed 18 partners and 9 European countries. And it has started in June 2021, and it will stop next year at the end of May next year. So in this slide, I will give you a very brief overview of the project because there is many aspects in this project. So I will try to give you a very brief overview. And so I think you can consider that there is 4 levels of action in Women. And the main level, of course, is on quantitative genetics. And there are 3 work packages allocated to that use quantitative genetics. So the VP3 that will work on the climate change and how to select animals that will be more thermal resistant in the future. So this is the main goal of the VP3. Then you have the VP4 that is mainly for genetic diversity. And in this VP4, we have also planned to resequence and to produce new genome assembly, but I will come back on this later. Then we have another level that is the genome editing level, the VP5. And in this level, we have 2 practical examples, but not in bovine, in small ruminant, one in goat and one in sheep. And I will come back also on this. Then we have 2 work packages, VP6 and VP7, that are dedicated to epigenetics. And so the aim here is to try to see by evaluating the epigenetics levels of the animal could help to select these animals in addition to the genetic evaluation. And thereafter, we have a 4th level that is the social aspect that is led by VP2 and VP9. And so we have had a multi-actor engagement, and the aim of the social aspect is to try to define a room of acceptability, what could be acceptable and what should not be acceptable. So next slide. So on this slide that is a little bit heavy, I have tried to summarize where we are actually in this 4 levels. So for the quantitative genetic, for quantitative genetic, for this aspect, until now, the VP3 that is dedicated of heat resistance is close to the end. And so VP3 have integrate the thermal and humidity index, the THI. So here integrate THI data with genotype and phenotype data that have been produced during the many years, during around 10 years, at the beginning of the genomic selection. And so they have crossed this data and they will propose now how to take into account intolerance in the selection scheme. For the VP4, VP4 have worked on genetic diversity, and they will propose now how to manage genetic drift, inbreeding depression, rare allele, and also genetic load. And so, as I told you, there is also the production of new genome assembly for 5 local breeds that are in progress. And I will come on this local breed on the next slide. For genome editing, no, sorry, yeah. For the genome editing aspect, I will speak mainly of the two examples, practical examples. So we have produce, we have in progress and produce pair and pay knockout goats, French goat, Alpine goats that should be normally resistant to prion disease. So we have obtained a founder for animal by genome editing. And actually, we have heterozygous animals, and they are currently bred in order to obtain F2 knockout animal and to precisely phenotype this animal. On another part, on another hand, a part that is led by the Rosin Institute. At the Rosin Institute, they have obtained founder prolactin receptor chip. So this founder carries a SLIC mutations that give a short hair on the animal, and so the animal are more resistant to heat. And so the founder for this SLIC mutation, a mutation firstly described in bovine, has been modelized in sheep, and these first animal have been obtained, and actually they are close to obtain F1 animals that will be phenotyped, and to see if the sheep are also resistant, more resistant to heat stress. For epigenetic, I think that our major contribution is that now we have a bovine-specific epichip that have been constructed by Illumina, and that is available since 1 year for the Rumigen consortium, but that will be available for everyone this year. So for the Rumigen consortium have tested these new tools. The technical development has been done, and actually epigenotyping of thousands of bovine are in progress, and I hope that we will see some preliminary result very soon in our next meeting. So to try to see if the fact to study the epigenetics in addition to genetics could improve the predictability of the selection scheme. And for the societal aspect, so I already told you that the final aim is to construct a room of acceptance. And so a first room of acceptance has been constructed, but with the consortium. And then 4 breeding scenarios have been built also and proposed to citizens. And this scenario has been used as a basis for a big consultation, a citizen consultation that was around a 2-hour survey with which has concerned 100 citizens in 8 European countries, so in total 800 of citizens for Europe. So my last slide is just to try to see how to interact and in between these different projects. And so I think that Rumigen already integrate different research area, and so it seems to match perfectly with the EcoGen mission. For the cross-project synergy, I think we have produced some interesting things that could be used by other scientists in other projects. For example, I think that the bovine epichip will be used worldwide. And the genome-edited animals that I have presented, the PNP knockout goat and the slick sheep could also be used for new projects, for example, looking to the microbiota of these animals could be an idea. And the new genome assembly for the 5 bovine breeds will also be certainly interesting for other scientists. So the 5 breeds are Abondance, Tarentaise, Vosgienne, MRY, and Norwegian wet cattle. And I hope that the new selection schemes that we will able to propose for bovine could be also inspiring for the other species. So the 2 questions I think that we could perhaps discuss thereafter is how to integrate microbiota data into selection scheme, but it's the same kind of questions that Diego already asked. And I see a paper, but I think it's for a future. It's only perspective. What about editing the genome of microbiota in order to act on gas emission, for example? So I thank you very much, and I let the floor to.
Yes, thank you very much, Eric. We managed to present the PPT, so that's perfect. Thank you very much. I will stop sharing my screen now and give the floor to Frédérique Pitel, who will present Geronimo. So let's move a bit to monogastric sector. And we can see your screen, Frédérique. So the floor is
okay?
Yeah, it's perfect.
Yeah.
Thank you.
Okay, perfect. Thank you very much. And thank you for the organization of this interesting webinar. So I'm Frédérique Pitel. I'm coordinating the Geronimo program with Tatiana Zerjal from INRAE also. And Geronimo stands for Genome and Epigenome Enabled Breeding in Monogastrics. You will see that we are a sister program of RumiGen. Oops. Yes. As we were talking about rumen microbiome and intestinal ecosystem, etc., and as because I feel that things are quite dark these days, I just wanted to start with a flower, just to talk about epigenetics, to illustrate epigenetics, because this is the main point of Geronimo. Epigenetics being the molecular marks that are appended to the DNA to change phenotype without changing the DNA sequence. And this is a nice example of such a change with the DNA methylation of one specific gene, which changes the phenotype without changing the DNA sequence. So this is the main idea of Geronimo. We know that the phenotype are controlled by genetics through DNA sequence variation and gene expression. And but we also know now that these epigenetic modifications, and Eric said that also, are important for building the phenotypes. And what is of interest for us is that the environment can trigger part of these epigenetic modifications. So in Geronimo, we have the same context as the other ones. So we have to move toward a more sustainable model because of all the current constraints, including environmental constraints, and I'm afraid it will not improve much. But in Geronimo, we are using omics technologies, such as the other ones, such as genotyping, epigenotyping, etc., to analyze both genetic and non-genetic mechanisms controlling traits, meaning the characteristics of animals, such as production traits, both in quantity and quality, efficiency, productive longevity, fertility, resilience, animal welfare and health, what we called efficient livestock production traits. And the final aim is to bring genome and epigenome enabled selection methods in, this time it's in monogastrics. We are working in chicken and pigs in the context of the Farm to Fork initiative. We have 22 partners from 11 countries, and I just wanted to highlight that we have of course universities, but also private companies and professional associations, because we are a multi-actor project, and because we are scientists, we needed experts in project management and innovation management. So we built this program around this idea, with two first work packages that aim to identify the underlying biological mechanisms affecting trait variation. In work package 1, we have big animal designs for our commercial partners that are phenotyped, genotyped, epigenotyped, etc., to really understand these mechanisms. And in work package 2, we are analyzing different environments, such as transport and rearing conditions, or feed quality, the temperature, the age of the animal, just to see how the environment can modify phenotype and how we can include that in the future selection models. In work package 3, such as in the Remigen, we propose strategies to optimize the conservation of genetic and epigenetic diversity by using especially local breeds. And in work package 4, thanks to all the phenotype, genotype, epigenetic data that we are producing in the other work packages, the idea is to develop methods to improve selection strategies that are integrating non-genetic components, in addition, of course, to the genetic. We also have a work package around ethical and societal dimensions regarding the application of genomic innovations to animal breeding. And in this work package, we'll also have a pilot study of epigenome editing. And finally, of course, we have the communication and dissemination work package. And here, I just wanted to highlight a few points that are really related, I think, to ecogene, which is the fact that we are bringing big omics analysis from different species in different environments. For example, in pigs, we have outdoor or indoor breeding with the test of two different diets with high or low proteins. It's the same in chicken with two different diets and chicken submitted to heat stress or controls that we are comparing. And in pig, we are using samples from a previous project of pigs that have been reared in a tropical area or in temperate area. And we have also metagenomes for microbiotes for these animals, and we are trying to integrate this metagenome plus epigenome, genome, and of course the phenotypes. Among the few results that we have now, we know that the maternal environment has a significant effect on the offspring semen production in pig. And we also have performed GWAS, that stands for Genome-Wide Association Studies. It means that we have detected genomic regions that control the variability of daily gain and back fat thickness in 3 pig breeds. We also have a quite original study on 15 generations of selection in pig. What we wanted to see was if we know we select genomic regions, so we have allelic frequency that are evolving along the generation with more or less selection. And we wanted to see how the epigenome was evolving with this selection. And we observed some regions with the same the same with similar patterns of evolution during the selection, while we observe other sites with different pattern of evolution, meaning that we may select differentially genetic and epigenetic. And yeah, one question we address in Geronimo is the length of the career of laying hens, because we know that the laying rate, the number of eggs that the laying hens lays, is decreasing with the age of the individual in weeks here. And the idea is to obtain many data on many samples from a commercial line of laying hens. So we obtain, again, genotype, phenotype, and epigenotypes to understand the way the number of eggs is reducing, and to try to improve this for a better sustainable model of laying hens. And we also have the genetic and epigenetic diversity analysis of several local breeds from France, Slovenia, Portugal, and many countries, with the idea of preserving this diversity. And about local breeds, there has been a survey with more than 500 local breeders that led to the production of a practice abstract named Local Breed, Local Voices, where the breeders are expressing their needs and for example, they perceive the risk around economic practices, and also on regulation, notably related to health. But for the social dimension, societal dimension, we also have surveys and interviews, as I have told before in work package 5. Our mathematical colleagues already tested the integration of non-genetic elements in their models. We also have a model with environment, which is a transmissibility model available. And my last questions are quite large. Will we be able to monitor the microbiome and or the epigenome using appropriate breeding systems? And how can we integrate all these disciplines of this data, these results, to improve sustainability? Quite large, sorry.
Perfect. Thank you very much, Frédérique. So we have now an overview now of many different species. I will give the floor to Antton Alberdi.
Good morning, everyone.
Yes, here you are.
Okay, I hope you can see the slide properly.
Perfect. Thank you.
Super. So I will give you an overview. Thank you. I will give you an overview of the project that I am coordinating from the University of Copenhagen. It is called 3D omics. We started back in 2021, and we are expected to finish it in December this year. And it is our project goes back to the rather fundamental stage of knowledge generation using I would say a radically different approach to analyze intestinal microbiomes, and then trying to bridge the gap between the understanding of how intestinal microbiomes affect animal breeding. So I will try to give you an overview of what we do and how we do this. Among these 13 partners distributed across 11 countries, we are 6 universities, 2 research centers, and 5 companies that are involved in the project. And the main idea within 3D omics is that the way microorganisms and their communities within animal intestines are analyzed and have been traditionally analyzed, or traditionally, say, in the last 15, 20 years, has been by studying bulk fecal or intestinal content samples where the spatial structure of the communities is completely lost. If you look at the left side, let's say that the conventional omics takes this approach where bacteria and other microorganisms are just loose in a sample with no spatial structure. But we know, based on microscopy data and other sources, that this is not the truth. In reality, the bacteria are structured in a specific spatial way, and we expect, based on the knowledge we have about general ecology, that the positioning with respect to each other, the positioning with respect to the epithelial layer, the mucosal barrier, the fecal pellet or the feed pellets, etc., can be very different. So when we came out with this idea of creating a completely different way to characterize microbial communities, we created this project, 3D omics, and what we have been trying to do in the last 3 years has been to implement this approach. This is a very simplified overview of how we have been working. So we have run experimental trials as regular trials with chickens and with pigs, and I will give you an overview of what were the questions that we were trying to answer, and we are trying to answer. So the main distinction has been that once we pulled the animals and we got the intestinal samples, we did not analyze those as a bulk sample. Instead, we embedded the intestinal samples, and then we cryo-sliced the intestinal tissue into this kind of very thin layers that take the entire circumference of the intestine. And then we used a laser microdissection microscope to get extremely tiny samples that are spatially referenced. So we have harvested thousands and thousands of samples that contain each about between 5 and 20 picograms of DNA, mostly bacterial DNA. And we have built sequencing libraries, and we are generating metabolomic data and other type of omic layers to characterize microbial communities at their scale, at the micron scale. So if you follow the scheme, then for the microdissection, we go to the omic profiling, where we divide this and we do different type of analysis to finally end up doing a spatial analysis that hopefully will give us a much higher resolution to be able to identify which are the relevant interactions between microbes and animals that are affecting the performance indicators of the animals, but also other kind of traits related to the sustainability, to the welfare of the animals, etc., etc. Just to give you a better overview of what I am talking about, on the left picture, you can see the laser microdissection microscope, which is a custom-made setup that we established in 2022 here in our department at the University of Copenhagen. And what we can do with this machine is to sample this kind of tissue samples. So if we go to the next slide, we see that the circumference of this tissue sample is about 1 centimeter of diameter. And then you might be able to discern that there are some small circular holes in the digesta. So hopefully you can see that if your screen is big enough. And there you can see that those holes are the samples that we are actually analyzing. So each of these holes has a specific spatial location, so we can know which microbial community has been characterized from which intestinal section specifically. And if we zoom in and do a fluorescence microscopy imaging of the microbial community, only I'm highlighting a few of those taxa, we can see that this is more or less the image that we would be obtaining. So we have many bacteria there that have, some are closer to each other in space, some are farther away from each other, but we can recreate a community that is specific to this location. And the diameter of each sample is more or less 50 micrometers. So we know that we can go slightly lower or higher in resolution, so the diameter will be lower. But for now, we are sticking to this 5,000 square microns that the samples usually are between 50 micrometers of diameter and 80 micrometers of diameter. And then we have been generating a lot of data from these spatially referenced libraries, and then validating all this information with fluorescence in situ hybridization imaging data. So what you can see on the top right is the fish data, where you have a lot of the bacteria, so not all of them, because we have only created probes for targeting the 15 most abundant taxa. And then we have used that for validating whether the reconstruction that we do of the microbial communities is close enough to what we observed from the fish data. I must say that the two types of data are not generated from the same sample. So we generate data from adjacent samples. So some degree of variability is expected. But as you can see on the bottom right chart, the main taxa at the genus level is match more or less. So we are quite happy to see that using metagenomic data generation at the very, very fine scale, we were able to recover the same signal that the fish imaging is giving us. The main benefit is that while using fish imaging, we can capture 15 different genera of bacteria using what we call microscale spatial metabolomics or metagenomics. We can cover hundreds and hundreds, and we can reconstruct their nearly entire genomes, and we can do functional analysis on knowing which bacteria strains with which type of genes are located in which location. And this is what we are using right now to identify new interactions or potential interactions that can be derived from this co-localization of different bacteria in different intestinal sections in different species. This was all about the methodology, and this is something that we expect to publish soon. We are in the very final stages of preparing the first manuscript that will describe all this new methodology. And what we are doing is then to use this new methodology in both poultry and swine. And what we have done is to run 3 different challenge experiments in poultry, one with adenovirus viruses, another one with Salmonella bacterium, another one with Histomonas protozoa. So what we have tried to see is how the interaction of the spatial distribution of bacteria and the different pathogens or zoonotic agents are affecting the animal performance, and also trying to understand why, for instance, in the case of Histomonas, is affecting us in a certain way the turkeys, while it is affecting in a completely different way chickens. So all the 3 experiments on poultry or trials have been focused on health, while the challenges we tackled in the swine model has been related to nutrition. So we have conducted experiments with prebiotic fibers and trying to understand how the location of the different fibers was affecting the spatial arrangement of the bacteria, and then how, depending on where the different bacteria would end up rearranging, how this would be affecting the performance of the swine. And in the other experiment, our focus has been on nitrogen digestibility and trying to understand how animals with different breeding scores were addressing different challenges in terms of dietary deprivation of nitrogen. Using all this information, we are conducting a lot of different studies, but at the same time, we are also putting all this publicly available for the rest of the community. If you visit our GitHub repository there, you will find a lot of data, but also a lot of bioinformatic tools that we have developed for analyzing all this type of data. And pretty soon, we will also have available the database that will enable anyone to browse through the different experiments and get these spatial overviews of the different samples and the community compositions in space. And at the same time, we are also depositing all the information, not just the nucleic acid sequencing data, but also the imaging data and the metabolomic data, etc., within this umbrella accession of this 3D Omics bio project. So in the future, everyone will be able to gather all this information we have generated in 3D Omics through this way. And just to finish with my final slides, I have added, as requested, 2 questions, and one would be for all of you. So what do you anticipate microscale processes could affect the functioning of the microbial systems that you work with? Not sure whether you have ever thought about this, but there might be the future potential to implement all this technology in completely different systems to address completely different questions. And we would really like to talk to people to see where they see this potential, and hopefully in the future pursue other projects that using this technology we can address other challenges. So thank you so much for your attention, and I hope I was in time.
Perfectly. And we have a question from David in the chat, and you have even 30 seconds to respond because a very simple question. If you have used rumen epithelial samples in your project.
No, yet. No, yet. The project was on monogastrics, so it's been just poultry and swine. But in the future, it will be really cool to implement this in ruminants as well.
Okay, super. Thank you, Antton. You were perfectly on time.
You're welcome.
So next speaker is David to talk about livestock, last project of this EcoGen joint initiative. David, I can see you, and you can share the screen when no, you can already share the screen, and the floor is yours. So it's David Yáñez from CSIC in Spain. We'll present you the last project, and after we will have roundtable. So you can start already to put questions in the chat. Yeah, you can see your screen, David. Go ahead, please.
Great, thank you, Anna. Thank you, Antton, for a very nice presentation, very interesting. Good afternoon, everyone, or good morning, or good evening, wherever you are. Well, I'd like to start thanking Anna and FAF for inviting us to be part of EcoGen cluster and also contributing today to this first event. So I'm presenting livestock, which is a Horizon Europe funded project. The title is Facilitating Innovations for Resilient Livestock Farming Systems. It started in 2022, so we are nearly halfway through the project now, 5 years project. And it's got a very broad and ambitious objective, which is to evaluate mobilize the adoption of a range of different innovative practices that can be applied at different scales, not only animal, but also herd, farm, etc., with a dual objective. So it's going through or towards mitigation and adaptation. So one is trying to reduce greenhouse gases emissions from livestock farming systems, including beef, dairy, cattle, and pigs. And on the other hand, towards adaptation, so increasing the capacity of those systems to deal with climate change impacts. So we're playing here with a complexity of a range of different practices, and you will see some examples later, but also towards mitigation and adaptation, and at the same time covering two very, very different animal species as cattle and pigs. So we have 37 partners from Europe and Australia, from 13 countries. Out of those 37, 16 partners are livestock industry partners, and you have a photo here of the consortium at our last General Assembly meeting in Edinburgh. So to address these very ambitious objectives, and I will now very quickly show you the structure of the project so you can understand how we are organized to trying to address these objectives. We have 3 groups of researchers or 3 work packages testing a range of different innovations that cover feeding, breeding, and farm management, and you will see some examples later on. Then the results of these tests, some of them will be then assessed for the socioeconomic and environmental impact in another work package, and also not only them individually, but in combination, some of them will be selected to redesign some systems at regional or European level to trying to be within the planetary boundaries. So we are testing innovations at different levels, but then those innovations will be then tested not only on the technical side, but also on the environmental and socioeconomic side, and then trying to see what would happen if we combine them at different scales. So this massive block of innovation testing and assessment of the impacts also interact with other components of the project. On the top, continuous and very important engagement with farmers and the livestock industry. And you will see some examples later on in which we basically use some innovations or some practices that are already being implemented by some stakeholders, and we go through the process of implementing those practices towards either mitigation or adaptation in a very regional or in a very specific regional context. And then at the bottom we have another work package, another group of colleagues working on the more the policy aspect. So what would be necessary to make these innovations implementation possible? And then, of course, we have a communication dissemination work package that it's actually strongly linked to the Ecogene cluster. So we take a look at this part of the project. So the kind of innovations or the practices that we are testing, obviously they are not things that probably you don't know. On the feeding side, we're working on different aspects. We are assessing the impact of using different type of feeds, alternative feeds, some of them byproducts, but also insects, etc., trying to reduce the carbon footprint of animal feeding. For that, we are also in strong collaboration with the Global Feed Life Cycle Assessment Institute, which provides one of the probably most relevant databases for carbon footprint associated to feed production. We're also working in different environments for grasslands in Scotland and in Northern Ireland and in Switzerland to trying to understand better the potential of multi-sward grasslands, but also how they can adapt to future climate change scenarios. We're also working on feed additives and the impact on microbiome and rumen fermentation with the aim of reducing methane emissions. And in this particular project, what we're working on is the combination of feed additives that have different mode of action and see how this combination can be either additive or synergistic. And we have so far achieved the 80% reduction of methane production by combining those type of feed additives. We have recently also published a special issue on feed additives for methane mitigation with 6 papers covering the whole range of phases needed for development and implementing anti-methanogenic feed additives. So you can have access to those papers in the project website. And we're also working on a range of different diets to try to help a different production system to deal with heat stress events. If we go to breeding, which is probably what is most relevant for the connection with all the Ecogene projects, we're working again on mitigation and adaptation. On mitigation, we are using one of the largest, if not the largest, database of phenotypes of animals for methane production in dairy cattle with records, individual records from Netherlands, Spain, and Poland, which are also associated to microbiome data. And the idea is to analyze the correlation between those traits, but also across countries to try to establish a common database that can be used for further developing the breeding programs. On the adaptation side, we're working again in cattle and in pigs. And in cattle, we are trying to understand the response of animals to different heat stress events. And for that, we are using mid-infrared records of 724 cows combined with respiration rate at different thermal situations. We're also working on a similar aspect in beef cattle, working on respiration rate at different thermal neutrality and thermal stress situations. And on pigs, we are using mostly not only metadata from different trials, but also metabolomic profiles from a large and small cohorts of animals using cosmopolitan and local breeds, in this case in Italy, to try to see if we are able to discriminate when the animal get to a heat load, but also most importantly, whether we can identify molecules that can be relevant or can identify animals that are either tolerant or sensitive to different heat stress conditions. This is the work that is being conducted now, led by the University of Bologna, which have already published a couple of papers on this, and the papers are available on the project website. On the third component of the testing innovation, farm management, this is a very broad group of experiments covering farm technology and farm management in different systems. And you have some examples there in pig, cattle, the manure management, agroforestry system to try to increase the capacity to capture carbon in the soil. And in some cases, the results from applying these technologies in farms will be also processed using artificial intelligence to try to develop tools to better predict animal behavior, animal welfare etc. And this is led by the University of Valencia, and it's bringing lots of interesting results as well. So if we go now again to the main project structure, and we focus on the assessment of the different impacts, then we are working on further developing LCA, life cycle assessment, improving the ability to assess the impact on different systems by also using different functional units, and also, as you know, the global warming potential debate about the best units and that we can use, and we are trying different possibilities, but we are also assessing the capacity of some systems to be resilient, and that's quite let's say challenging, because the assessment of the resilience is not something that is already in place, and we are working on that, and we are also trying to assess what would happen if we combine some of the innovations I mentioned before in terms of circularity. For animal welfare, we have identified 5 different locations that cover different climatic conditions in Europe, and together with the Potsdam Institute for Climate Change in Germany, we have predicted the situation in terms of no heat stress or heavy heat stress for animals in those locations. And in those locations, we are running now animal trials. So it'd be interesting to see whether we can develop protocols to better test animal welfare in those locations. As I said before, we are also running different case studies, and the information is available on the project website. We are running 13 case studies testing how the implementation of different practices towards either mitigation or adaptation took place, what challenges the stakeholders, mostly farmers, faced when implementing or trying to implement these practices. And again, those cover feeding, breeding, and farm technology and farm management. Right, and I know that Anna is desperate to start the discussion. So just bring a couple of questions related to our activity that maybe can be used for the discussion. First, can we assess the impact of combining innovations? We are testing different kind of innovations across these different and other European projects, and we should not consider them in isolation. And also something that has been also discussed and presented, trying to test and develop region-specific innovations, because one solution doesn't fit all situations. And with that said, again, thank you, Anna, and I'm very happy to take any questions.
Yes, thank you, David. There is a question for you that is linked a bit to your first question, and that is combining technologies. Well, we can make progress, and there is one specific question about methane, of course, in the chat, saying that the reducing methane by 80% is great. So any insight yet on the impact of this on rumen function and animal performance? And do we need, and that's where it's linked to your first question, do we need to now target the rest, the last 20%, and which other technologies? I mean, you mentioned a bit breeding, of course, but this combination, has responded to your own question and how the combination of those technologies we can achieve more progress. And then after I will give room and tackle all the questions that are in there.
But very quickly on the reduction of methane, we know the 30% reduction of 35% reduction, it's okay without impairing rumen fermentation, and that's the case of bovine and other feed additives. But if the idea we developing in the project is if we combine feed additives that target other parts of the whole metabolic process of producing methane, and that's how we have achieved the 80% reduction in vitro. And we are now in Rusitech that works fine. So there's no excess of hydrogen, because one of the actions that we have developed is to try to capture the excess of hydrogen that when methanogens are inhibited. And yeah, the idea now is to go into the in vivo trials and see whether that's still possible, or whether the 80% maybe result into 50 or 60% in vivo conditions.
Yeah, and I think that's responded at the same time to the second question in the chat from Karel de Graaf. They're saying, hey, this is we do that. We're still digital. Sorry, muting people. If you are not talking, please can you mute yourself? And that said, will still cause digest favor as well. So that's the point of passing from in vitro to in vivo thing, I suppose.
Yeah, the overall fermentation is not affected, but yeah, that has to be confirmed in vivo trials.
Exactly. We have 2 hands raised. So as far as we have questions from the audience, I will start skipping your questions between you, between the 5 coordinators that were raised. Rick Brunches, I guess. Or something
Pretty
like
good.
this. Sorry, my
Thanks,
Dutch.
David. It was super interesting. Could you say a little bit more about the combination of your 80% mitigation at the moment? And also, as you just said about because I wasn't sure, I only just heard in your response it was in vitro. I thought it was in vivo, which is clearly a bit of a difference. But just, yeah, about the combination, what you used, if you can say anything about that.
Yeah, of course. Yeah, we can identify 3 main steps in methane production. So first is the degradation of carbohydrates, then is the direction of the hydrogen, the electrons somewhere, and then the methane production. So the idea is to use additives that work in the 3 phases, trying to lower hydrogen production, but also include alternative hydrogen acceptors that can capture the hydrogen that is produced in excess when the third one, methanogens, are inhibited. So and those at the moment we have used monensin for the first one. We know that monensin is not allowed in the EU, but it's a good model because it's consistent in the chemistry, etc. Nitrate and 3-NOP.
Thanks very much.
Yeah, and we tested over the 3 weeks incubation periods in Rusitech, and yeah, the production is 80%. But as I said before, yeah, that needs to be further tested in vivo.
Okay, a second hand raised, Juliaxis, please.
Hello, everyone. Thank you to all the presenters. I have a specific question for David. David, how we can do, because I see that one possibility is to join a dataset from project with common interest, and that is the case from HoloRuminant and your project in the particular part of the dataset you have for methane and genotypic information, but also the dataset we have for the same, or trying to answer the same question.
I think this question is probably not only for me, but for all coordinators. And this is something we've been discussing before. I think we need to devote some time and effort to trying to join forces across the different projects. And one of them is that, is a very good example, is how can we incorporate all the databases from one project to another to make that more powerful. And I think that's possible, but and probably obviously FAB here is a very good example of coordination, but also I think that should be done in connection with the European Commission.
Yeah, absolutely.
Thank you.
So that's just to remind you, this is the first webinar of a series in which we will tackle specific topics. So I think Diego and other coordinators, Antton, Frédérique, Eric, do you want to also to jump on this question? I think it will be interesting. Yes?
Concerning the Juliaxis question, I think we should be open to share our data that is in the spirit of everybody, but not in the facts. Sometimes because it takes time and how we can share that without changing a lot of things in our data sets, and that's the difficult part, I think. So we should work also on how to make common protocols for metadata, so they can be explored by other people. And one of the parts also that is also associated with this EcoGene project is that we share our data in the Eurofund platform.
Yes.
So a lot, I don't know if every project, but some of our projects are committed to put our sequence data, at least, in the Eurofam platform. So that's a way to do that. But we all know that just sequencing data or metaproteomics data or whatever omics data is not of any value if you don't have the associated metadata to explore that. Otherwise, you cannot explain the changes.
Yes, thank you, Diego. Indeed, the Eurofunk initiative is a concept on development in research infrastructure, so it's not already in place, but they are trying to reflect on what you are saying, how to share data, how to build infrastructures, not only in terms of facilities, but also data data sets and so on. Is anybody else having a question? I don't see questions in the chat.
Perhaps I can
okay, Antton,
follow
you
up
can jump
on this.
Okay.
No, it's just to give you a case example of what I think it is. It's a good initiative, and this does not relate to the current European project that we are running through the omics, but the previous one, which was called HoloFood. And what we did was to create a repository that I hope you can see when I share the screen, which is called HoloFood Data. And this repository puts together all the different omic layers that we have deposited in the different databases so that people can easily browse through it, and then can also make sense to the metadata with respect to all the experimental designs and the questions that we created, et cetera, et cetera. So if we put everything in the public repositories, it might be fetchable, but it is usually very difficult to make sense to anything. So I think this is a good example that other people could also try mirror, because this is also open access. So the way the database is created, et cetera, can be reproduced in other projects. So just so you know that this resource existed, and it is fully based on fetching data that are stored in the EBI's, European Bioinformatics Institute's different databases, and then putting everything together so that you can browse easily and say, okay, if you go over to this animal, then fetch all the imaging, metabolomics, sequencing data from that same individual. And then it is far easier for people then to reuse this data. So it's just an example.
Okay, thank you, Antton. Very useful. There is someone asking, what about the European Nucleode Archive?
But that's exactly what this is based on.
Okay.
But
okay.
the problem that we have with the INRAE, the European Nucleode Archive, is that you might archive everything there, and we must archive everything there. But then I think we need to make an extra effort to make all this information more reachable for people. And this is what, in my opinion, the European Nucleotide Archive is a little bit deficient. The way the information is displayed, of course, you can get everything programmatically, and it is really fine. But you really need to have a very good overview of the project in order to be able to catch everything. So this is why I was mentioning this.
Okay, super. Thank you very much. Any other questions? Anybody aiming to jump on previous presentations? Ruminant, Rumigen, etc. So perhaps then we can take some of the questions you were asking yourself. There was some of them talking about how to go to, more on implementation side, and so you were discussing about these data sets, but after how we integrate this information into selection schemes, into breeding programs. That was one question that was raised by 3 of you, if I remember well. So perhaps people from the private sector is here around. I mean, perhaps not, I'm sure. Or perhaps other coordinators within the 5 projects would like to respond. Frédérique, David, you are a lot of in contact with breeding companies.
Well, yeah, I can maybe make a comment. I'm not an expert on breeding, but I think it's very important to work hand in hand with the breeding companies from the beginning of the project. I think that's essential. If that collaboration starts later on, then well, the success is doubtful. So I think it's really important on this project to bring them right from the beginning, so that they are co-developing and understanding the process and also making decisions as researchers. Otherwise, yeah.
Even before the project starts, I mean, during the proposal creation, absolutely. You have many of you have to, Frédérique, sorry.
Yeah, sorry, I just find a hand.
Go ahead, yes.
Yeah, just about the applicability of our results in real life. Private partners are really interesting. If we manage to include a microbiome or epigenome in the model, of course, but we need to have tools that allows them to genotype, epigenotype, microbiotype, not too in a not too expensive way. You see what I mean? It's not applicable if it's too expensive. And I think for example, the epi chip from Eric, when less expensive, can be used on a high level, high throughput. But it's really important for us to know that it won't be used if it's too expensive, of course.
Yeah, absolutely. And that comes also to a topic that you were addressing, many of you, is that you are addressing all this research to breeds that are everywhere, cosmopolitan breed, but also local breeds. And when it comes to implementing local breeds or smaller populations, that's this price or the accessibility of technology is more difficult for a question of resources, of course. Not only, but mainly. Eric, you opened your camera. Do you wanted to add something?
Just to say that, Frédérique, I agree completely with Frédérique. There is a big problem of price, but we take it into account for the EP chip in Morgavi, because we have built a very small chip, not so small, more than 40,000 mark, methylation mark, but small if you look at all the possible mark. But the main our main aim was the price of the chip, and because we hope that the chip will be able to be used directly on the real life. We will see. It depends if it works or.
Okay, thank you. Well, it's 1 minute. It's 12:30 in my computer at least. So I would like to thank the 5 coordinators and all the audience for your attendance. We are going to put in the chat. There is already a web page with more details about the 5 projects, so the cluster of Ecogen. Of course, there is the 5 projects website. You know now the coordinators, you can be in touch with them. Again, this was the episode 1 of a series of webinars of the cluster. The second one will be resilience and adaptation, and the third one, microbiome and livestock health. So yeah, follow us in the different social medias, and if you have subscribed to newsletter and so, you will be informed. So we will tackle other topics in the upcoming webinars. And thank you again for your attendance and your engagement during the roundtable. Thank you very much.
Thank you.
Bye.
Thank you. Bye.
Thank you.
Thank you.
Bye.
Bye.
Thank you.
Thank you. Bye.